Dr. Sharon Goldberg is an integrative internal medicine practitioner in New Mexico. She is currently Associate Professor of Medicine, Community Faculty at the University of New Mexico School of Medicine.
Good morning, everyone. I'm very excited to be here talking about the bacterial effects of electromagnetic fields. When I initially proposed this topic, I thought that it was going to be a simple talk about antibiotic resistance. But once I started researching it, I realized that the story is actually much more complex and much more interesting than I had initially thought. So, what is the story? I read a lot of papers on this topic, and to sum up the key issues, I put together this iceberg diagram. And essentially, the way that I look at it is you've got the top half and the bottom half. So what you see at the top of the iceberg are what I call the visible effects. If you were to go and do a literature search right now, you would come up with two types of articles. One type of article showing antibiotic resistance induced by EMF exposure, and another group of articles showing antibiotic-like effects. As you go further down the iceberg, you see effects that are less visible from a literature search. For instance, effects on soil bacteria. There were a few papers that I found on that. Effects on bacteria that live in and on our food supply, on plants and animals. Then going further down, you see effects, these are direct effects of EMF exposure on the human microbiome. And finally, you see indirect effects on the microbiome. For instance, there are effects of whole body EMF exposure causing stress and then causing, for instance, disruption of sleep-wake cycle, hormones, etc. So, in 2019, the CDC published this report, Antibiotic Resistance in the United States. And I think we can all agree that antibiotic resistance is one of the greatest public health challenges of our time. It costs us a fortune. It leads to millions of additional hospital days. But what's less obvious is that antibiotic failure threatens the way that modern medicine is practiced. Because without antibiotics, chemotherapy, organ transplants, things like hemodialysis and joint replacements, we really wouldn't be able to do them successfully. So just a few statistics from that CDC report. If you do the math, you can see that you've got over 3 million infections per year and nearly 50,000 deaths per year. So it's clearly a very big problem. So what influences bacterial responses to electromagnetic fields? And by the way, this talk is a radio frequency focused talk. The papers that I'm going to show you are all RF, but please keep in mind that also magnetic fields and lower frequency EMFs also are proven to have effects on bacteria. They're just not included in this talk. So there are two sets of factors to consider. First, you've got the exposure conditions. So things like wavelength, frequency, intensity, how many repeated irradiation, etc. These are related to the EMF itself. The second set of factors are bacterial factors. So we're talking about gram-negative versus gram-positive bacteria, spore forming. Are they anaerobic or aerobic, growth medium? I'm not going to read the list, but you can see it's all related to the bacteria. And so which bacteria have been studied? You'll be surprised to see that some of our major pathogens have been shown they become antibiotic resistant when exposed to radiofrequency radiation. And just to show you from that list, sorry, I should go back. It includes Pseudomonas aeruginosa and Staph aureus, two major human pathogens, which, by the way, made the serious threat level criteria in that CDC report that I showed you at the beginning of the talk. So they're serious pathogens. So let's look at some of the research. I'm focusing on, when I show you these papers, I'm focusing on the organisms that they've been tested and the conclusions. Some of them we'll go through quickly. This is a 2019 paper by Moavedi et al. That examined the effect of a mobile phone simulator on Pseudomonas and Staph aureus. And here's what they found. Pseudomonas aeruginosa became resistant to all antibiotics after 24 hours exposure to 900 megahertz versus unexposed controls. Staph aureus became resistant to levofloxacin, tetracycline, and trimethoprim sulfa, but it remained sensitive to piperacillin, ampicillin, and ceftriaxone. Here's another one, 2017, Taheri and colleagues. They looked at Listeria monocytogenes and E. coli after exposure to 900, so they exposed the bacteria to either 900 megahertz phone simulator or a 2.4 gigahertz Wi-Fi router for 12 hours. And these were their conclusions. Exposure to RF EMF within a narrow window of irradiation, in other words, an exposure window, makes microorganisms resistant to antibiotics. They go on, these findings may have serious implications for the management of serious infectious disease. Got one more, 2019, Saeed Salman and colleagues exposed E. coli, Staph aureus, and Staph epi to a Wi-Fi router for 24 or 48 hours. And here are their conclusions. Wi-Fi exposure acted on bacteria in a stressful manner by increasing antibiotic resistance and motility of E. coli, as well as enhancing biofilm formation ability in E. coli, Staph aureus, and Staph epi. So the bottom line of the paper, their conclusions, are that these findings may have implications for the management of serious diseases caused by these infectious bacteria. So as you can see, there's no shortage of research showing antibiotic resistance and from EMF exposure. And secondly, the authors they're clearly concerned about their findings. So I want to talk go a little bit into depth about this last paper, the Saeed Salman 2019. There's a lot to learn from this paper. So again, they looked at 3 different bacteria, E. coli, Staph aureus, and Staph epi. And the aims of the study were to figure out the effect of Wi-Fi on 3 endpoints: antibiotic resistance, cell metabolic activity, and ability to form biofilm. The exposure was Wi-Fi for 24 or 48 hours, and they did have controls kept in a Faraday bag. So the findings that they found, they found antibiotic resistance just in E. coli. They found increased motility also in just E. coli. But in all 3 organisms, they found an increase in biofilm formation. So what does this mean? Well, biofilm and motility are virulence factors, particularly for E. coli. So let's talk about that a little bit. Flagella, they permit motility and make it possible for E. coli to adhere to mucins, which are the major component of mucus that lines the GI tract. Other stressors, such as heat and acid, are known to promote motility. And so the bottom line is that increased motility of E. coli after radiofrequency exposure likely represents a strategy for pathogen survival. The second finding that they talk about was biofilm. So what is it? It's a protected mode of growth that allows cells to, number 1, survive in hostile environments, and number 2, disperse to colonize new niches. And so why do we care? Well, it plays a role in a wide variety of human infections, such as line infections, implants, bacteremia, and gum disease. So I've got one last article related to E. coli, and this is a gene expression study. The reason why I'm showing you this is because it really helps us understand the results of the previous studies. So here's what they did. It's 2019, Saeed Salman, the same group as the last article I showed you. Their aim was to investigate the changes in global transcriptome in E. coli after 5-hour exposure to a Wi-Fi router. So their conclusions, I'm going to tell you in simple terms, is that Wi-Fi, the exposure triggered differential expression of genes that promoted virulence and survival mechanisms. And their conclusion was that undervaluing the problem of telecommunications exposure could cause further rise in infectious diseases or their complications. And this is a graphic from that paper that shows you nicely what happened. So you can see the E. coli in the center of the screen. And in green, you can see the genes that were upregulated. And they all have to do with stress response, DNA damage, and other virulence factors. The genes that were downregulated had to do with essentially having the the bacteria spend less energy on metabolism, and it went into a growth arrest and a dormant state to survive this stressful exposure. So here I've got an overview of the findings of this study. I'm not going to read it, but if anyone wants the full info on that study, it's right here. So to make sense of these studies, we need to talk a bit about antibiotic resistance mechanisms. Antibiotic resistance is either intrinsic to the organism or it's acquired. Acquired resistance occurs in two main ways: either acquisition of exogenous genes, such as from a plasmid, or spontaneous mutation of cellular genes, which happens to be extremely rare. There is one exception to this rule that's very relevant to our topic, and it's called the hypermutable state model. So certain strains of bacteria are called hypermutable strains, and it just so happens that E. coli, Staph aureus, and Pseudomonas aeruginosa are hypermutable strains. These types of strains can increase their rate of mutation up to 10,000 times during a prolonged non-lethal selection of microorganisms. So in other words, this is referred to as adaptive mutagenesis. And there's selection pressure on these bacteria, and they mutate at an extremely high rate. So some antibiotics, as well as certain electromagnetic fields, induce this SOS mutagenic response, which increases the rate of emergence of resistance to antibiotics. So we just covered non-lethal exposures, but what about lethal exposures? EMFs don't just induce antibiotic resistance. They also are found in the literature to induce antibiotic-like effects. So, and these are harnessed in industrial applications. For instance, EMFs are used to disinfect agricultural wastewater and used in the food industry to inactivate pathogenic microorganisms. So the question is, we are all being exposed to RF 24/7 now, with some exceptions, but most of us. And if ubiquitous RF exposure leads to population-wide antibiotic-like effects, even in a small percentage of these exposures, we really need to think about this closely, because it could be a big problem. And certain situations, we would expect it to be a particular problem, i.e. the soil where our food is grown, areas where meat and poultry are raised, and particularly the human microbiome. So I mentioned soil effects. I've got one paper here about EMF and soil bacteria. So this is a paper from 2018, Sharma and colleagues, the effect of mobile tower radiation on microbial diversity in soil and antibiotic resistance. So what they did was they took 4 soil samples taken from less than 50 meters from a cell tower and 4 control samples that were taken from greater than 300 meters away from a tower. What they found was that they found greater antibiotic resistance observed in microbes present in soil near base stations compared to controls. They also found a statistically significant difference in the pattern of antibiotic resistance found with nalidixic acid and cefepime. They concluded, our findings suggest that mobile tower radiation can significantly alter the vital systems in microbes and turn them multi-drug resistant. So shifting gears now, we all understand that the microbiome plays an important role in human health, but since we're talking about EMFs EMFs having antibiotic-like effects, we need to talk a bit about the human microbiome. So there's an estimated 500 to 1,000 species of bacteria that exist in the human body at any given time. Bacterial cells outnumber human cells by a factor of 10. And really, it's not just the gut flora. Changes in the microbiome and the microbial metabolome and the way they interact with immune, endocrine, and our nervous systems correlate with a wide array of illnesses, including inflammatory bowel disease like UC and Crohn's, major depression and autism, and other medical conditions such as cancer, obesity, and metabolic syndrome. So I've got 3 points to make on the microbiome. So most important is that the microbiome is not just about gut flora. The human microbiome is highly personalized, and in this diagram, you can see that different colors represent different species. The skin shows dramatic variation in microbiome composition and structure across different sites. In other words, you'll have different organisms on different areas of the skin, different organisms in the oral cavity, and all sorts of all over the body. So it's not just in the GI tract. And most important, the skin plays a significant role in shaping the individuality of the entire microbiome. Point number 2 is that the human microbiome is extremely diverse. So you can see that there are gram-positive organisms here, rods, cocci, spirochetes, gram-negatives, and then you have anaerobes and aerobes. So we understand that EMF effects are, certain strains are affected more than others, and because there's such diversity, this could lead to degradation of microbial ecology, a selective degradation of certain types of organisms. My third point about the microbiome is that the gut microbiome is highly influenced by diet, host, and environmental factors. So, for example, studies have shown that when a person makes a major dietary change, within 24 hours, you will see a change in the gut flora. The same thing goes for stress and sleep deprivation. So what it boils down to is that the gut flora can either be health promoting or it can be disease promoting. And to give you some examples of this, symbiotic effects of gut flora include host nutrition. For instance, certain bacterial byproducts help us digest foods that we eat, immune development, protection from pathogens. And to give you one example, short-chain fatty acids, they help maintain the colonic epithelium. They provide energy for the host and regulate the immune system. Conversely, some examples of dysbiotic effects, you can see that microbial metabolites can have toxic effects. They're not all beneficial effects. And an example would be antibiotics. We know that antibiotics disrupt microbial ecology, reduce the diversity of the GI and oral microbiome within days of administration. And complete recovery of the initial bacterial community is rarely achieved. I have one last slide before I conclude that sums up why we need EMF literate clinicians who consider the clinical relevance of EMF science. Historically, EMF science has been a mostly non-clinical field. So this is a 2013 study of Lactobacillus acidophilus exposed to millimeter waves. Their intervention was they exposed Lactobacillus to 51.8 and 53 gigahertz for 1 hour, plus or minus ceftazidime, compared to unexposed controls. Their conclusions: strong antibacterial effects were found on Lactobacillus acidophilus. Bactericidal effects were comparable to ceftazidime. And in case you're squinting and confused about this paper, yes, this is Lactobacillus acidophilus, as in yogurt and as in probiotics. So clearly, we need clinicians in this field to be thinking about the potential consequences of using EMF that could potentially harm bacteria, beneficial bacteria. So how do we tie this all together? If you only remember one thing from this talk, it's that EMFs act on bacteria and their hosts in a stressful manner. The stress leads to altered gene expression. There are direct effects of EMF exposure on bacteria, and they include, number 1, induction of antibiotic resistance, number 2, an increase in virulence factors such as biofilms and motility, and number 3, antimicrobial effects. There are secondary effects of EMF on the human microbiome, which include the downstream effects of stress, such as sleep, wake cycle disruption, metabolic disruption, and hormonal effects. So what I hope you take away from this talk is that this is a really important clinical issue. It requires further study, And thank you for being here at the conference. We need clinicians involved in thinking about these issues. Thank you.